In this video, I'll be reviewing a research article discussing the connection between transposable elements and gene regulation in mammalian genomes. The human genome possesses multiple different sections responsible for various roles associated with gene coding. Yet, despite almost half of the entire human genome consisting of small segments of DNA referred to as transposable elements, these DNA sequences were often overlooked and under-researched. The lack of focus on transposable elements came from the misplaced assumption that they played no role in aspects of gene regulation and expression. The research performed in this article largely centered around the impact of transposable elements on 3D chromatin structure and the subsequent effect these alterations would have on gene regulation. Existing primarily within transposable elements, the CTCF protein determines the limitations of chromatin looping in the chromosomes of mammals, with any modifications to this protein potentially leading to non-traditional 3D chromatin structure through chromatin loop divergence. This knowledge, along with the discovery that chromosome structure, influenced by chromatin looping, is heavily involved in phenotypic expression at a cellular level, provided evidence for a substantial link between transposable elements and gene expression. I found it quite interesting that these small, seemingly unimportant segments of DNA played such a crucial part in the expression of so many genes through their influence on the process of chromatin looping. Another point of personal interest outlined through this article is that the relationship between the CTCF protein and the chromatin structure of which it is responsible for is more heavily bonded than I had initially presumed. In cases when the function of CTCF is nullified through researchers performing protein knockdown, chromatin looping is heavily disrupted throughout the gene, leading to a massive divergence in 3D chromatin structure. This loop disruption directly altered gene expression to a certain extent, with it being assumed that this was caused by modifications to the enhancer-promoter region. This paper also briefly references the discovery of how transposable elements, colloquially known as jumping genes, were able to impact the phenotype that a corn plant expressed. The mention of this finding directly mirrors an area of learning that I covered in Gen 2052, in which the concept of specific transposable elements affecting gene excision repair was visualized through the alteration of gene expression within corn kernels. This particular example exhibited the ways in which transposable elements could play a role in modifying the pigment gene via differences in the location at which they were excised, with this leading to the consequential phenotype expression of either full pigment, no pigment, or an intermediate state. Overall, I felt that this article provided me with more extensive knowledge surrounding a learning area of Gen 2052 that I was immediately intrigued by. Research revolving around the different segments of the human genome, their associated roles, and the effects that arise when these sequences are altered is incredibly important, and there is still so much more to explore.